Welcome to the deep dive. Today, we're tackling something absolutely fundamental. We're going deep into the mechanism of how life, well, copies itself.
And, just as importantly, how it fixes the inevitable mistakes in that copy.
We are talking about DNA replication and the incredible world of DNA repair.
These processes are, they're the absolute bedrock of reproduction, not just for whole organisms, but for every single cell in your body. Our guide for this is Chapter 13, From Cell and Molecular Biology, Concepts and Experiments.
And this chapter starts right away with this amazing biological paradox, which the geneticist Richard Lewontin summed up so well.
Oh, it's a great quote.
Right. The DNA molecule has all the information to duplicate itself, but it can't actually do it. It's like a blueprint that can't build the house.
Exactly. It's completely inert in that sense. It needs a huge crew of cellular machinery.
Yeah.
Enzymes, clamps, motors, a whole repair team.
Yeah.
Just to carry out the instructions written in its own code.
So our mission today is to walk you through all of that. We're going to break down every major idea, every key experiment, and all that complex machinery from the chapter to make it as clear and accessible as possible.
Let's start at the very beginning, with the structure itself and how scientists first guessed it might copy.
So 1953, Watson and Crick figure out the double helix structure, and almost immediately they see a way it could replicate.
They did. The idea was just, it was inherent in the structure, because the two strands are complementary, A always with T, G with C.
And they're held together by hydrogen bonds, which are relatively weak.
Right, so you could, in theory, just pull them apart, like unzipping a zipper. And then each of those separated parental strands could serve as a perfect template for building a new partner strand.
Which led to the model they favored, called semiconservative replication.
A very descriptive name. Each new DNA duplex would be a hybrid. It would conserve one of the old parental strands and have one brand new daughter strand, half old, half new.
But, this was a totally unknown process, so the scientific community had to think about other ways it could possibly work.
Of course. And the first alternative was conservative replication.
So in that model, the parent molecule would open up, act as a template.
But then, somehow, the two original parental strands would find each other again and reform the original molecule,
leaving you with one DNA molecule that's the original parent, untouched, and a second molecule that is 100% brand new.
Exactly. And then there's a third sort of stranger idea.
Dispersive replication.
Right. And this was taken pretty seriously, especially by people like Max Delbruck. He was worried about the topology of it all.
The unwinding problem.
The unwinding problem. How do you unwind these incredibly long, intertwined strands without just breaking them? So the dispersive model suggested that maybe the parental strands get chopped up into fragments.
And then the new DNA is synthesized in pieces and sort of, shuffled in?
Yeah, creating these mosaic strands that are a mix of old and new segments all along their length.
Okay, so we have three really distinct hypotheses: semi-conservative, conservative, and dispersive. How on earth did they figure out which one was actually happening inside a bacterium?
Well, they needed a way to physically tell the old DNA apart from the new DNA. And this leads us to what is, just one of the most beautiful experiments in biology.
The Meselson-Stahl experiment, 1957.
Just brilliant. The core idea was labeling. They used two different isotopes of nitrogen, the common, light one, which is $14, and a heavier, rare isotope, $15.
And nitrogen is in the DNA basis, so this was a perfect tag.
Perfect. So first they grew E. coli bacteria for many, many generations in a medium where the only source of nitrogen was the heavy $15.
So eventually all the DNA and all the bacteria was heavy.
Every single strand. Then they did the key step. They took these bacteria and transferred them into a new medium that only had the light $14.
And then they just let them divide and replicate their DNA.
Exactly. And they took samples over time. To analyze them, they used a technique called equilibrium density gradient centrifugation.
Which sounds complicated, but the idea is pretty simple.
It is. You spin the DNA in a tube with a cesium chloride solution at super high speeds. A density gradient forms, and the DNA flutes to the level where its own density matches the solution's density.
So heavy DNA sinks lower, light DNA floats higher.
Precisely. And the results they got were just unambiguous.
So after one single generation of growth in the light medium, what did they see?
They saw only one band of DNA. And critically, that band was located exactly halfway between where the all-heavy DNA would be and where the all-light DNA would be.
An intermediate hybrid band.
A perfect hybrid.
Yeah.
And that single result right there immediately killed the conservative model.
Because if it were conservative, you'd expect two bands.
You'd have to. You'd have the original heavy parent molecule in one band and the brand new light daughter molecule in a separate higher band. They only saw one. So conservative replication was out.
Okay, but a hybrid band could still fit the dispersive model, where everything is a mix of old and new.
It could. So they had to let the bacteria divide one more time. The second generation.
What happened then?
That was the clincher. After the second generation, they no longer saw one band. They saw two distinct bands.
Two bands?
One was still at that hybrid intermediate position, and the other was a new band that was completely light, all $14.
And that is the absolute signature of semi-conservative replication.
It's the only way to get that result. The hybrid molecule from generation 1 unwound, its heavy strand templated a new light strand, making another hybrid molecule. But its light strand templated a new light strand.
Making a completely light molecule, hence the two bands. It's just so elegant.
It really is. And this wasn't just some weird thing that bacteria do.
No, it's universal. They showed the same thing in eukaryotes, in cultured mammalian cells.
A slightly different technique, but the same principle. They used a chemical called bromodeoxyuridine, or BRDU, which your cells will mistake for thymidine and build into DNA.
So if you look at the figure in the chapter, figure 13.4, you can see how this works with chromosomes. After one round of replication with BRDU, both of the sister chromatids in a chromosome are hybrids. They contain BRDU.
Right. But after the second round of replication, something interesting happens when you stain them. One of the chromatids is still a hybrid, with one old strand and one new BRDU strand, so it stains dark.
But the other chromatid is now made of two BRDU-containing strands. It's all new, so it stands light.
And seeing that consistent pattern of one dark and one light chromatid after two divisions proved it. It's semi-conservative, all the way from bacteria to us.
Okay, so we've established the what, it's semi-conservative. Now we have to get into the how. What is the actual machinery doing the copying?
And for that, we go back to bacteria, where all this was first figured out. I mean, it was a huge challenge. Replication is essential for life, so if you break a gene involved in it, the cell just dies.
You can't study a dead cell.
Exactly. So they got clever. They used two main approaches. The first was isolating temperature-sensitive mutants.
I love this strategy. It's so smart.
It's brilliant. You find a mutant that has a protein that works fine at a lower permissive temperature, but when you raise the heat just a little bit to the non-permissive temperature, protein unfolds and stops working.
So you can grow the cells normally, and then boom, you turn up the heat, and you can see exactly what part of the process breaks down. It's like having an on-off switch for a lethal mutation.
Precisely. And the second big approach was developing in vitro systems. Purifying all the individual proteins and then trying to rebuild the entire replication process in a test tube.
And through all that work, they found out that even a simple E. coli cell needs more than 30 different proteins working together just to copy its DNA.
It's a real machine, and it's a very organized process. In bacteria, with their circular chromosome, replication doesn't just start randomly.
Right. It starts at one specific place.
A single defined site called the origin, or orice, in E. coli. And from that spot, it proceeds bidirectionally.
Meaning two copying machines basically start there and move in opposite directions around the circle.
Yes, forming two replication forks that travel away from the origin until they eventually meet on the other side.
The second those forks start moving and pulling the strands apart, you run right into that problem Delbruck was worried about, the unwinding problem.
Topological stress. It's a massive mechanical issue.
The analogy is like taking a twisted rope. If you try to pull the two strands apart in the middle,
the part of the rope ahead of where you're pulling gets wound up even tighter. It gets super coiled.
And in DNA, that manifests as positive supercoils building up ahead of the replication fork. And if that tension isn't relieved, the whole process would just grind to a halt.
The fork would get physically stuck.
Mm-hmm.
So the cell needs a stress reliever. And it has one of the most incredible enzymes there is, DNA gyrase.
Which is a type of topoisomerase.
A type 2 topoisomerase, specifically. Its whole job is to travel ahead of the fork and fix this supercoiling problem.
And the way it does it is just, it's wild. It doesn't just untwist it.
No, it's much more dramatic. It performs a kind of molecular surgery, makes a temporary controlled double-stranded break. It cuts both strands of the DNA.
Whoa.
Then it passes another segment of the DNA right through that gap, which physically removes the supercoil.
And then it seals the break back up perfectly.
And then it reseals it. It's an ATP-dependent process, so it takes energy, but it's absolutely essential to allow the fork to keep moving. It's a controlled break to prevent a catastrophic one.
Okay, so gyrase clears the path. The strands are separated. Now finally, the enzymes that actually do the copying can get to work. The DNA polymerases.
And the story here starts with Arthur Kornberg, who first purified DNA polymerase, or poly, back in the 50s.
And he figured out the basic recipe for making DNA.
Right. You need a DNA template to read, you need the building blocks, all four deoxyribonucleoside triphosphates, or DNTPs, and you need energy.
But he also discovered two really critical limitations, two rules that this enzyme has to follow.
Yes, and these were huge clues. The first one is that I cannot initiate a new strand from scratch. It's not a starter.
It can only add nucleotides onto the end of a chain that's already there.
Specifically, onto the 3' hydroxyl terminus of an existing strand, which we call the primer. You can see this in figure 13.7. That 3' OH group is the non-negotiable handle it needs to grab onto.
Okay, so it needs a primer. What's the second rule?
The second rule is the directionality problem. All DNA polymerases, poly included, can only build a new strand in one direction, the 5' to 3' direction.
And this creates a huge paradox at the replication fork.
A massive one, because the two parental strands of the double helix are antiparallel. They run in opposite directions. So as the fork opens, the polymerase would have to move one way on one strand and the opposite way on the other strand.
But the enzyme can only go one way, 5' to 3'.
Exactly. It seemed impossible. And then, to top it all off, in 1969, a mutant E. coli was found that had almost no polymer activity, but it replicated its DNA just fine.
So, Paul, I wasn't the main replicator after all.
It got demoted. The real star of the show turned out to be a much rarer enzyme, DNA polymerase III, or Pol3. But, and this is the key, Polar follows the exact same rules. It needs a primer, and it only works 5' to 3'.
So the fundamental problem, the directionality paradox, was still there. How does the cell solve it?
The solution is just incredibly clever. It's a process called semidiscontinuous replication.
Okay, let's break that down.
The cell basically realizes that synthesizing in the 3' to 5' direction is impossible, so it just doesn't even try. It synthesizes both new strands in the correct 5' to 3' direction.
But how if they're on antiparallel templates?
By treating them differently. On one template strand, the polymerase can move continuously toward the replication fork as it opens. This is the easy one. We call it the leading strand.
Just needs one camera at the start, and then it can go.
Uninterrupted. But on the other template, the lagging strand, the polymerase has to move away from the replication fork to maintain that 5' to 3' direction.
So it can't be continuous.
It can't. It has to wait for the fork to open up a stretch of template. Then it synthesizes a short fragment backwards, away from the fork. Then it has to jump ahead to where the fork has opened up more template and do it again.
So one strand is made continuously and the other is made discontinuously in little pieces. Semi-discontinuous.
Precisely. And we have concrete proof of these pieces, which we call Okazaki fragments, thanks to Reiji Okazaki's experiments.
We use pulse labeling, right?
Yes. Very short bursts of radioactive thymidine, just a few seconds. And in that tiny window, he found that all the radioactivity was incorporated into these small DNA fragments, about 1,000 to 2,000 nucleotides long in bacteria.
But if you waited a bit longer, a chase period,
the radioactivity would move from those small fragments into much, much larger continuous strands of DNA.
Proving that the small pieces were being stitched together into a complete strand.
And that stitching is done by an enzyme called DNA ligase, which seals the final gap in the sugar phosphate backbone.
Okay, but this brings us back to the other problem. Pol3 can't start a chain. So where do the primers come from for the leading strand and for every single one of those Okazaki fragments?
The cell solves this with a different kind of nucleic acid. It uses RNA primers.
Ah, so it's a temporary starter.
Exactly. An enzyme called primase, which is a type of RNA polymerase, builds a short little stretch of RNA, maybe 10 nucleotides long, right on the DNA template.
And because it's an RNA polymerase, it can start a chain from scratch.
It can. So the primase lays down that RNA primer, which provides the crucial 3' OH group that Pol3 needs to get started. Pol3 then takes over and starts adding DNA nucleotides.
So the replication fork is a seriously busy place. You've got all these different players.
It's a factory. So let's look at the key components you see in figure 13.12. First, you need to unwind the DNA. That's helicase. In E. coli, it's a protein called DNA.
It's a ring-shaped protein that uses ATP to just power down the DNA and rip the strands apart?
Yes, it moves along the lagging strand template, and once the strands are separated, they want to snap right back together.
So you need something to hold them apart.
That's the job of single-stranded DNA-binding proteins, or SSBs. They just coat the exposed single strands and keep them extended and protected.
And then you have the primase, which coordinates with the helicase.
Right. They form a unit called the primosome. As the helicase unwinds, the primase periodically hops onto the lagging strand to lay down a new RNA primer for the next Okazaki fragment.
Okay, this brings us to the big choreography question. You have two polymer enzymes. One is cruising along the leading strand toward the fork. The other has to go backwards on the lagging strand. How do they stay coordinated as part of a single machine?
This is where the trombone model comes in. It's a brilliant explanation shown in figure 13.13. It proposes that the two pol-3 polymerases are physically linked together in one big complex, the replisome.
But how can the lagging strand polymerase move away from the fork if it's tied to a machine moving toward the fork?
It does it by looping the DNA. The lagging strand template is actually looped back on itself 180 degrees.
So the template is fed through the enzyme backwards, basically.
Exactly. This reorients the lagging strand so that, from the perspective of their replisome, both polymerases are moving in the same physical direction, even though they are copying antiparallel strands.
That's amazing. So as it makes an Okazaki fragment, that loop of DNA gets bigger.
It gets bigger and bigger. Then, when the polymerase finishes a fragment, it lets go, the loop collapses, and it grabs onto the next primer closer to the fork, and a new loop starts to form.
It grows and shrinks like the slide on a trombone.
Hence the name. It's how the cell keeps everything efficient and coordinated at the fork.
And for all this to work, Paul III has to be incredibly fast, and it has to stay stuck to the DNA.
That's the property we call processivity. And it's provided by an amazing accessory protein, the beta clamp.
Which is shaped like a donut.
Its shape is its function. It forms a ring that completely encircles the DNA, and it tethers the pol-3 enzyme to the template. Without the clamp, pol-3 would fall off after just a few bases. With the clamp, it can go for thousands.
But if it's a closed ring, how does it get onto the DNA in the middle of a strand?
It needs help. There's another machine called the clamp loader. It uses the energy of ATP to grab a beta clamp, pry it open, slip it over the DNA at a primer junction, and then snap it shut.
A whole other energy-requiring step just to get the clamp in place.
For every single Okazaki fragment, yes. And just to bring it full circle, what happens to those RNA primers?
They have to be removed.
And that's where our old friend pol-3 goes back. It has a special activity that allows it to chew away the RNA primer from one end, while simultaneously filling the gap with DNA from the other.
So it does the cleanup and patching, which leads us to the most important question of all.
Yeah.
How does this whole system make sure the copy is perfect?
This is all about fidelity. I mean, the accuracy is just staggering. The final error rate is less than one mistake in a billion nucleotides.
Which is incredible because the polymerase itself isn't quite that good on its own.
No, the raw error rate is more like 1 in 100,000.
Yeah.
So the cell uses a 3-tiered system to achieve that near perfection.
What's the first line of defense?
The first line is just accurate nucleotide selection, and it's all about geometry. The active site of the polymerase is shaped to perfectly fit a correct Watson-Crick base pair. An AT pair and a GC pair have the exact same dimensions.
But a mismatch, like a G trying to pair with a T, would be a different shape. It wouldn't fit right.
It wouldn't. And that bad fit prevents the enzyme from closing its fingers around the nucleotide to catalyze the reaction. It's a process called induced fit. This initial geometric check filters out most errors.
But a few still slip through, so what's the second layer?
The second layer is immediate proofreading. This is a function of the polymerase's 3' to 5' exonuclease activity.
So it's a built-in delete key.
That's exactly right. If the polymerase accidentally adds the wrong base, the geometry of that mismatched end is wrong. This causes the enzyme to stall.
And then what happens?
That frayed, incorrect end of the new strand is moved from the polymerization site into a second active site on the enzyme, the exonuclease site.
Where it gets snipped off.
The wrong base is removed. The strand is now correct again. It moves back to the polymerization site, and synthesis continues. This proofreading step improves fidelity by another 1,000-fold or so.
And the third and final layer is a separate system. Post-replicative mismatch repair, which we'll get to later. It's the final cleanup crew.
But before we get to repair, we have to scale up. We've been talking about E. coli with its single chromosome. What about eukaryotes? Our genomes are 1,000 times bigger.
And our polymerases are actually slower. So how do we copy all that DNA in just a few hours during S phase?
We do it by starting in many places at once. Instead of one origin, a human cell has tens of thousands of origins. The genome is copied in many small segments called replicons.
Which means the cell needs exquisite control over when and where all those origins fire.
Absolutely. And the timing isn't random. Active open regions of chromatin, the euchromatin, tend to get replicated early in S phase.
While the tightly packed silent stuff, the heterochromatin, gets copied late.
Exactly. It's an epigenetic level of control.
So how does the cell make sure that each of those thousands of origins fires exactly once, and only once, per cell cycle?
This is the once per cycle rule, and it's controlled by a two-step licensing process. It all starts with the origin recognition complex, or ORC. This is a protein complex that binds to the origins and acts as a landing pad.
And it stays there all the time.
It stays there. Then in the G1 phase of the cell cycle, the origin gets licensed. This means another set of proteins, the MCM proteins, which are the eukaryotic helicase, get loaded onto the ORC. This forms the pre-replication complex, or pre-RC.
So a licensed origin is ready to go.
It's armed and ready. Then when S phase begins, protein kinases, especially a cyclin-dependent kinase or CDK, get activated. They trigger the licensed origins to fire and start replication.
And what stops them from firing again?
That same CDK activity. While CDK is active, from S phase all the way through mitosis, it actually prevents the loading of any new MCM proteins. It blocks the formation of new pre-RCs.
So you can't relicense an origin until the cell has divided and CDK levels drop again in the next G1. Brilliant.
It guarantees everything is copied just once.
Now, what about the eukaryotic fork itself? The players are a bit different.
They are. We use a trio of main polymerases, polymerase alpha, delta, and epsilon.
What do they all do?
Pol alpha works with primase. It's the initiator. It makes the RNA primer and then adds the first 20 or so DNA bases.
And then it hands off the job.
Yes, in a process called polymerase switching. The current model is that pol epsilon takes over and does the continuous synthesis of the leading strand.
And pol delta does the lagging strand.
That's right. And both pol delta and epsilon need their own sliding clamp for processivity.
And the eukaryotic clamp is called PCNA.
Yes, proliferating cell nuclear antigen. It serves the same function as the beta clamp in bacteria, but it's also more than that. It acts as a kind of molecular tool belt.
What do you mean by that?
It not only holds the polymerase on, but it also recruits a whole host of other proteins to the fork enzymes for repair, for chromatin remodeling. It's a central coordinating hub.
And the cleanup of the primers on the lagging strand is also a little different, right?
It is. Because the Okazaki fragments are so much shorter, only about 150 bases, pol delta actually pushes the previous RNA primer out of the way, creating a little flap.
And that flap gets cut off
by a special nucleus called FEN1. Then DNA ligase comes in and seals the nick.
One last thing on replication. All of this is happening on DNA that's wrapped around histones in nucleosomes.
A huge challenge. The machinery has to disassemble the nucleosomes ahead of the fork and reassemble them immediately behind it.
And this is critical for epigenetic inheritance. The cell has to copy not just the DNA sequence, but the patterns of histone modifications and DNA methylation that tell genes whether to be on or off.
It's a whole other layer of information. For DNA methylation, an enzyme called DNMT1 follows the fork and copies the methylation pattern from the old parental strand onto the new daughter strand.
And for histones, it seems to be a system of positive feedback. An old modified histone helps recruit enzymes that place the same modification on the new histones nearby.
It's an incredibly complex and active process to ensure that when a cell divides, the daughter cells aren't just genetically identical, but epigenetically identical too.
So if replication is about making a perfect copy, repair is about what happens when the DNA gets damaged after it's been copied.
And it gets damaged constantly. The chapter gives a staggering number. A single one of your cells can suffer about 10,000 spontaneous base damages or lesions every single day.
From what?
All sorts of things. Reactive chemicals inside the cell, just simple thermal energy, and of course external things like UV radiation from the sun.
UV light causes pyrimidine dimers, where two adjacent bases on the same strand get stuck together.
Exactly. And if that damage isn't fixed, it can lead to a permanent mutation the next time the cell tries to replicate that DNA.
So these repair systems are our constant defense against cancer and aging.
They are. Let's start with a system that deals with bulky, helix-distorting problems, like those pyrimidine dimers. That's Nucleotide Excision Repair, or NER.
And NER has two subpathways, right? The cell prioritizes.
It does. There's the transcription-coupled pathway. This one gives priority to fixing damage on the template strand of genes that are actively being transcribed.
How does it know?
An RNA polymerase literally gets stuck when it hits the lesion. That stall is the signal to bring in the NER machinery. It's a fast lane for repair of the most important parts of the genome.
And the other
Is
pathway.
the global genomic pathway. It's slower, and it scans and repairs the DNA everywhere else.
But the core mechanism is the same for both, this sort of cut-and-patch approach.
Yes. Once the lesion is recognized, helicases, including two that are actually part of the transcription factor, TFIIH, unwind the DNA around the damage.
Then what?
Endonucleases come in and make two cuts in the damaged strand, one on either side of the lesion,
They bracket the damage.
and they snip out that whole segment, maybe 30 nucleotides long. That leaves a gap, which is then filled in by a DNA polymerase using the opposite strand as a perfect template. And finally, DNA ligase seals it up.
Okay, so NER handles the big stuff. What about smaller problems, like a single chemically altered base?
For that, you have base excision repair, or BER. This system targets things like a cytosine that's accidentally turned into a uracil, or bases damaged by oxidation.
And it's initiated by a whole family of enzymes called DNA glycosylases.
And each glycosylase is a specialist. It roams the DNA looking for one specific type of damage. When it finds it, it doesn't cut the backbone. It just cleaves the bond holding the bad base to the sugar.
It just pops the base right off.
It does. The chapter shows this amazing mechanism for one of them where it literally flips the nucleotide completely out of the helix and into its active site to inspect it.
That's incredible.
It's how it checks for damage with such high precision.
And this actually explains why our DNA uses thymine instead of uracil.
It does, because cytosine can spontaneously change into uracil. If DNA normally had uracil, the repair system wouldn't know if a uracil it found was supposed to be there, or if it was a damaged cytosine. By using thymine, any uracil found in DNA is automatically flagged as damage.
Smart. So after the glycosylase removes the base, what happens to that empty spot?
An AP endonuclease cuts the backbone at the empty site. A DNA polymerase comes in, removes the rest of the sugar phosphate, fills the one base gap, and a ligatus seals it. It's a much more targeted repair than NER.
Okay, what about the errors that replication itself makes? The mismatches that proofreading missed?
That's the job of mismatch repair, or MMR. Its biggest challenge is figuring out which the two mismatched bases is the wrong one.
It has to know which was the original template strand and which is the new error-containing strand.
Exactly. If it fixes the template, it makes mutation permanent. In E. coli, it tells the difference because the old parental strands have methyl tags on them, and the new strands don't yet. The MMR system is directed to repair the unmethylated strand.
And finally, the most dangerous damage of all, double-strand breaks.
DSBs, caused by things like X-rays or gamma rays. This is when the chromosome is literally snapped in two. It's a catastrophic injury for the cell.
And there are two main ways to fix it. One is fast and dirty.
That's non-homologous end joining, or NHEJ. It's the dominant pathway in our cells. A set of proteins, including one called Ku, basically grab the two broken ends and just stick them back together.
Why is it dirty or error-prone?
Because the ends are often damaged. In the process of ramming them together, a few nucleotides often get lost or added. It saves the chromosome from being lost entirely, but it almost always creates a small mutation at the repair site.
And the cleaner, more accurate option
Is homologous recombination, or HR. This pathway uses an undamaged identical copy of the broken DNA as a template to ensure a perfect repair.
And that perfect copy is the sister chromatid.
Exactly, which means HR can only happen after DNA replication has occurred, in the late S or G2 phase of the cell cycle, when that sister chromatid is available. It's much more restricted, but it's error-free.
So what happens when these systems fail? The chapter's human perspective section gives some really stark examples.
It really does. The classic one is xeroderma pigmentosum, or XP. These patients have a defect in the nucleotide excision repair pathway.
So they can't fix the damage from UV light.
They can't. They have extreme sensitivity to the sun and a massively increased risk of skin cancer. It's a direct link between a repair pathway and a disease.
But then there's a related disease, Cockayne syndrome, or CS. They're also sensitive to light, but they have developmental problems and don't get cancer at the same rate. Why the difference?
It's because in CS, the defect is specifically in the transcription-coupled part of NER. The global repair pathway still works, so their overall mutation rate isn't as high. The symptoms are thought to be from the constant disruption of transcription in essential genes, leading to cell death and developmental issues.
It's incredible how a subtle difference in which part of a pathway fails leads to a totally different disease.
And there's a hypothesis that connects these ideas to aging and cancer.
Right. The idea is that repair defects that lead to a high mutation rate, where cells survive but are full of errors like in XP,
That leads to cancer.
but defects that lead to widespread cell death because the cells can't tolerate the damage and undergo apoptosis.
That leads to symptoms of accelerated aging because you're losing cells and depleting tissues. It's a choice between mutation or death.
Now, what happens when the replication fork is moving along and it runs into a lesion that hasn't been repaired? Does it just stop?
A stalled fork is a disaster, so the cell has a last-ditch mechanism called translesion synthesis, or TLS.
This is a damage bypass system.
It is. The regular high-fidelity polymerase stalls. That signals for a special low-fidelity polymerase to be swapped in temporarily.
So it's calling in a specialist.
A specialist in dealing with damage. A great example is polymerase eta. When it encounters a thymidine dimer from UV damage, it has the unique ability to correctly insert two adenines opposite the dimer, allowing replication to continue past the block.
And a defect in polyemia is what causes the XP variant form of the disease.
Exactly. Their NER is fine, but they can't do this bypass cleanly, which makes them sensitive to UV light.
So what's the catch with these TLS polymerases? Why doesn't the cell just use them all the time?
Because they're designed to be sloppy. They have a much more open, spacious active site that can accommodate bulky, damaged bases that the normal polymerases would reject.
But that flexibility means they make more mistakes.
A lot more. And crucially, they have no proofreading exonuclease activity. They are highly error-prone. The cell uses them only as an emergency measure. It's a calculated risk. Better to introduce a potential point mutation than to have a complete collapse of the replication fork.
All of this just underscores how direct the line is from these molecular processes to human health.
It's completely direct. UV exposure and NER failure lead to skin cancer. Defects in mismatch repair are a major cause of hereditary colon cancer. Even things like radon gas in your basement can cause double-strand breaks in your lungs and lead to lung cancer. Understanding these pathways is fundamental to understanding and fighting these diseases.
This has been an incredible deep dive. Let's try to quickly recap the biggest takeaways from all this complexity.
I think the first one is foundational. Replication is semiconservative. And because of the antiparallel nature of DNA, it has to be semidiscontinuous, using a continuous leading strand and a fragmented lagging strand.
Second is the machinery itself. The replism is this beautifully coordinated complex of helicases, clamps, and polymerases, all optimized for speed and, critically, for fidelity.
And that fidelity is multi-layered. You have the initial base selection, then the immediate 3' to 5' proofreading, and finally, the backup repair systems.
And third, the genome is under constant assault, and the cell has a whole toolkit of repair systems, NER, BER, MMR, and the double-strand break repair pathways, each specialized for a different kind of damage. Failure in any of them has severe consequences.
Absolutely.
So for final thought, I want to go back to the speed of the bacterial replism. You said it copies 1,000 nucleotides a second.
That's right.
Just think about the choreography that demands. In 1 second, the helicase has unwound 1,000 base pairs, the gyrase ahead of it has cut and resealed the DNA multiple times, the primase has laid down a primer, and the lagging strand polymerase has synthesized an entire Okazaki fragment, proofread it, detached, and is now being reloaded onto the next primer.
The coordination is almost unimaginable.
And it brings up a question we haven't really focused on. The sheer energetic cost of all this.
It's enormous. Every single one of those major steps, gyrase cutting, helicase unwinding, the clamp loader opening the clamp, the polymerization itself, it all burns ATP.
So this process of flawlessly copying and maintaining the genome isn't just a marvel of information science and mechanics.
It's also probably the single most energetically expensive process the cell ever undertakes, which just tells you how incredibly important it is to get it right. It's a constant, costly, high-stakes operation that defines life itself.
A stunning piece of engineering inside every living thing. Thank you for joining us for this deep dive into the mechanics of life.